Hi everyone, this is our lecture on chapter 10, which is meiosis and sexual reproduction. So the last chapter, chapter 9, we talked about mitosis, right, which is just cell division of your somatic cells, right? Meiosis, and in mitosis, you produce two identical daughter cells, right? So remember, obviously there's lots of mitosis that goes on during development, right? So if we're talking about starting from a single zygote to then forming an embryo and a fetus, right? Lots and lots of mitosis and cell division that goes on there. Also during growth and repair. When we talk about meiosis, particularly, we're going to talk about the formation of gametes, which or sex cells. Okay, and so this is only going to happen, in the ovaries and in the testes in terms of producing egg cells and sperm cells. Okay, and the process, there are a lot of similarities between meiosis and mitosis, and we'll talk about that. But there are also differences, and in the main difference is basically the purpose and the types of cells that are produced. Right. In meiosis, again, those cells will not be identical. Okay. In fact, it's super important that there's lots of genetic variation that's introduced, and we'll talk about that. Okay. So, in terms of the outline overview of meiosis, I just mentioned this genetic variation. We'll talk about the specific phases of meiosis. This is also in your lab that you are doing the meiosis and mitosis lab. You also go over the phases of meiosis as well in terms of what's happening and being able to recognize those phases, from pictures. And then we'll talk a little bit about meiosis compared to mitosis, like I said. Then just overall kind of talking about the cycle of life and where, meiosis fits in and where mitosis fits in, okay? And then the last part of the chapter talks about changes in chromosome number and structure and different diseases or disorders that might lead to, okay? All right, so I guess there's a lot going on in this chapter. All right, so the first thing that we're going to talk about is just meiosis in general, right? An overview. And so I did mention this in the introduction that it does bring about a huge amount of diversity, right? Of genetic diversity, which is obviously super important. If it didn't, we would all walk around being the same person genetically. So obviously we know that's not true. There are, in fact, more than 70 trillion different genetic combinations possible from the mating of two individuals. So that's just one mating, right? 70 trillion different genetic combinations. So obviously lots of diversity, lots of genetic variation. Males, females differ in the way they form gametes, right? We're talking about egg cell production versus sperm cell production. There is differences, both involve meiosis, but there's differences there. So it mentions here that sperm production begins at puberty, right, in males, but in the female, the process of producing eggs starts before birth and then obviously ends at menopause. Okay, so slightly different. And again, like I said, there are some variations in the process of oogenesis, which is the production of egg cells, and spermatogenesis. We'll talk about that briefly, but I don't really hold you guys responsible to really knowing the details or the subtle details between those two, just because there's a lot of other stuff going on in this chapter. Okay, so in terms of, again, meiosis, I said, it's a special type of cell division that only occurs in your gametes, right, in your sex cells. It's used only for sexual reproduction. It is not used for growth and repair or anything like that. Like I said, only in the ovaries and in the testes, this is going to occur. So just like we saw with mitosis, right, you still have chromosomes that would be replicated in S phase, right? And then they're halved prior to fertilization. So you're going to end up with gametes that have a haploid number, right? So remember, we talked about diploid and haploid before. So parents are diploid 2N, but when you're going to go through meiosis and produce those gametes or those sex cells, the egg cell or the sperm cell will be haploid. Okay, so it only has one set of chromosomes, not two. All right. So again, reminder: haploid cells contain a single set of chromosomes, like I just said. Right. If there were no reduction of chromosomes in meiosis, the number of chromosomes would just keep doubling each generation. And right, and instead of having two sets of chromosomes, you would have some ridiculous number of sets of chromosomes, and that doesn't work out. And so you'll even see at the end of the chapter when we talk about, some some diseases or disorders that are associated with an extra chromosome, right? Or lacking a chromosome, just one extra chromosome is going to cause an issue. So obviously you can't keep increasing the number of chromosomes. It says here, gametes, right? The sperm and the egg cell will fuse during fertilization and they will form the zygote, right? So you have two haploid gametes, egg and sperm cell, that then fuse together to form a diploid zygote, okay? So obviously you can see why it's important that the gametes are haploid, okay? When they fuse together, they will become diploid. All right. So it says here, if events in meiosis go wrong, gametes can contain the wrong number of chromosomes. So again, that leads you to talking about diseases or disorders that are characterized by the wrong number of chromosomes, right? So these are not diseases that are caused by a mutation in a single gene. These are problems where you have an extra chromosome or part of the chromosome broke off and things like that. So we will talk about that at the end of the chapter. Okay. Here again, in diploid body cells, chromosomes occur in pairs, right? Remember 2n, right? 2 pairs of 23. 23 different types of chromosomes. Again, 2 chromosomes of each in diploid. We say that those chromosomes of the same type are said to be homologous. I'm going to show you a picture in a second. Okay, so homologous chromosomes are homologs. That's what this says here. So they have the same length. Their centromeres are positioned in the same place, right? One came from the father, which is the paternal homolog, and one from the mother, the maternal homolog, okay? And when you stain them and look at them, they show similar banding patterns, okay? So what they mean by that is looking at this, right? See, these are the banding patterns. So again, you can see 23 chromosomes, right? This is the sex chromosomes. You have 2 sets of 23, right? One paternal and one maternal. And if you look at this, it's called a karyotype. If you look at this, notice, right, what the chromosomes look like. They just have one single arm because they're not duplicated, right? Remember I told you when you talk about a duplicated chromosome, it's that X, that sort of X characteristic shape that you think of. Those are the duplicated chromosomes, right? But when we're looking at normal just chromosomes, when they're not duplicated, you have, again, one arm. So again, one maternal, one paternal, right? Chromosome 2, a maternal copy, a paternal copy. So each one of these chromosomes contains the same genes, and we're going to talk about this a little bit more, but they contain variations of genes. So think about, your mother has blue eyes and your father has brown eyes. Your mother has brown hair, your father has blonde hair, right? There's genes that control hair color, genes that control eye color, but you have variations in what type of gene. Okay, we'll leave it at that. And there's a term for that, which is alleles. And again, we're going to bring up these terms later on. We're also going to talk about those terms even more in the next chapter, chapter 11. Okay. So again, a reminder, we talk about chromosome, we look at it just a karyotype, which shows all the chromosomes. So this is a normal karyotype, right? And you can see there's no Y chromosome here. They have 2 X's, right? So this is a female. And again, these are what the chromosomes look like. One paternal, one maternal copy, 2 sets. Okay, they're not duplicated. Here now, this is showing you once they're duplicated. Remember, the chromosomes are going to have to be duplicated in S phase, right, before the cell goes through meiosis, right, or before it goes through mitosis. And so now, again, if you look at the homologous pair, now you can see that these are duplicated. Okay, so whenever you see 2 arms or this sort of X kind of shape, you know that these are duplicated. And then they'll have sister chromatids, right? Remember we talked about that in mitosis as well. So again, the pair, the homologous pair is this, the paternal and the maternal, and that's what this is showing you here, okay? All right, so again, this may take a little bit of time to kind of go through. So remember, there are animations that I posted on Moodle. There's also other videos that you guys could look up if the sort of sister chromatid and the homologous pair situation is confusing you. You also obviously ask me about it at our next class meeting and I can go over it again, okay? But we want to know the difference, super important. Okay, oops. So I probably already said some of the things that are on this slide, and I apologize for there being so many words, right? I already said this. Homologous chromosomes have genes controlling the same trait at the same position, okay? A maternal copy and a paternal copy. Many genes exist in several variant forms, obviously, in a population. Okay. Otherwise, again, we would all have brown hair and we would all have brown eyes and there would be no variation. Okay. Those are just easy kind of things to conceptualize. Homologous copies of a gene may encode identical or different genetic information, right? It depends. Okay. So again, we're going to call those variants alleles, like I mentioned, right? So the form that you have on the maternal chromosome versus the form that you have on the paternal chromosome of that gene, right, is called an allele. Those are different. It's the same gene, but different variants of that gene. Okay. All right. So it says here, an individual may have identical alleles, right? They can be the same for a specific gene on both homologs, right? And that would mean that you're homozygous for the trait. And again, remember, we're going to talk about these things. Chapter 11 really focuses on this type of genetics, okay? A maternal allele that differs from the corresponding paternal allele, that would mean that you're heterozygous for the trait, right? So it gives you an example here, a gene coding for short fingers on one homolog and a gene coding for long fingers at the same location on the other. That would be heterozygous, okay? If you are homozygous, you would have the gene coding for short fingers on both the maternal and the, right? If you're homozygous, both maternal or paternal, all right? So again, these terms in terms of homozygous and heterozygous and homozygous recessive, homozygous dominant, we are going to talk about these in the next chapter, in chapter 11. Okay, so you don't have to worry about grasping everything here because again, we will focus on that in the next chapter. Okay, we're just bringing out some of these terms, introducing them, but again, re-emphasizing them later on. Okay. So in terms of meiosis and the process of meiosis, right, we're going to go over this first. There's two sets of nuclear division. So when we talked about mitosis, there was just one set of division. Okay, so in meiosis, it's meiosis 1 and meiosis 2. And like I said, you're going to be working through this a little bit on your own when you're filling out that PDF of the lab. Okay, again, this is just something that it's very straightforward. You can look at pictures and see what's happening at each one and do it on your own. I don't need to tell you every single thing. So I'll go over it here, but I don't know. I may not include every single detail that's written on the slides with the pictures, okay? Because like I said, you're going to work through that on your own. In fact, it's probably more useful for you to work through it on your own, write down what's happening at each stage and be able to recognize those stages, okay? All right, so meiosis 1 in general. So now this is just general in terms of what's happening. We will go through each stage of meiosis 1 and each stage of meiosis 2 as well, like I said, okay? Chromosomes are replicated prior to meiosis 1, okay? So we said that, right? Chromosomes still have to be replicated during S phase, okay? And then you'll have those sister chromatids, okay? Then what happens is those homologous chromosomes will actually line up with each other in something called synapsis. And at that point, they'll actually recombine and exchange genetic material. So that will introduce genetic variability, which is what we want, right? So that's what this is saying. So that will happen, right? The exchange of genetic information. Then those homologous pairs will align themselves at the metaphase plate, okay? And then during meiosis 1, the homologous pairs will separate, okay? So the pairs will separate, they'll still have their sister chromatids, okay? So each daughter cell will receive one duplicated chromosome from each pair. So important that one cell gets one chromosome and the other one gets the other one so that you make sure you have all 23 chromosomes in each one of those cells, right? If that doesn't happen, that's where you can get defects in terms of the number of chromosomes right, that exists. Okay, and like I said, we'll talk about that at the end of the chapter. Then in meiosis 2, no DNA replication will occur after meiosis 1. Now the sister chromatids will actually separate from one another. So meiosis 2 looks a lot more like mitosis, okay, in terms of those steps. And then basically, so each one of those daughter cells that are produced in meiosis 1 will then form, you'll get 2 daughter cells from there. So you're going to end up with 4 daughter cells after meiosis 1 and meiosis 2, okay? Right, so now they're going to contain 1 daughter chromosome from each pair, okay? And then at that point, you only have a single chromatid. And again, the daughter cells will be haploid, okay? So the daughter cells after meiosis 1 are haploid as well, because you only have one copy of each chromosome. Even though it's duplicated, it's just one copy of each chromosome. Okay, so let's look at some pictures in terms of this overview. Again, this is just an overview, and then we will go through the stages in a little more detail. I guess I can go back this way. All right, so this is showing you a cell here. 2n equals 4. So again, in our cells, we have 23 chromosomes, okay? And 2n would equal 46, right? What this is just showing you is here is just two different, so you have essentially two sets of 2 here, okay? So this is showing you a homologous pair here. Here's another homologous pair, okay? Paternal and maternal. So just so you kind of know what this is denoting. So obviously we sort of oversimplify so that we can see what's happening with those chromosomes. All right. Now, DNA gets replicated, right, during S phase, just like we saw with mitosis. So now, what do you notice? These chromosomes only had one arm here, right? Now they're duplicated. They have sister chromatids. So we can tell that they're duplicated because they have that characteristic X shape, and they have 2 sister chromatids, okay? So each one of these was duplicated. Now those are exact copies, right? When these sister chromatids are exact copies of one another at this point. All right. So again, right, 2n equals 4, but they're just duplicated here. Okay, so if we move on, so that's now what this is showing you that these guys are going to line up with one another. Okay, these homologous pairs. Okay, again, these are the same chromosome, one paternal, one maternal. They're going to line up with each other, and at this point, they're going to exchange genetic information. Okay, it's not really showing you that. We'll focus on that in the next couple slides, but that's the synapsis part. Okay, it's showing that they're going to line up. All right, at the end of meiosis 1, right, those homologous, the homologous pairs will separate, right? So you can see now in each one of these daughter cells, you have so in this case, you have the paternal copy of chromosome 1, and you have the maternal copy of chromosome 2, right? Here, you have the maternal copy of chromosome 1, and you have the paternal copy of chromosome 2, okay? But in each case, you have chromosome 1 and you have chromosome 2, okay? It just happens to be that you have different variations there. All right, so that's the end of meiosis 1, and then if we move into meiosis 2, each one of these daughter cells will then go through another division, right? And this, meiosis 2, will look very similar to mitosis. All right, so the sister chromatids separate, and now again, you're ending up with 4 daughter cells, okay? All of which are haploid. Okay, they're only, now if you, right, they only have one set of each chromosome, right? You don't have two pairs, you have one set here. Remember, we started off here with 2 pairs, right, with a pair of each chromosome. Here, you only have one of each chromosome, okay? So diploid in this case, right? This is diploid. Again, if you notice here, now these are haploid, n equals 2. So wherever you see 2n, that's diploid. Wherever you see n equals haploid, okay? Again, remember in our cells, you would have 2n equals 46, and down here, n equals 23, okay? All right, so now let's talk more specifically about that genetic variation that's occurring, okay? So I mentioned the synapsis part, so we're gonna talk a little bit more about that, and then also about something else, okay, that we also kind of saw as well. Okay, so again, genetic variation is essential for a species to evolve and to be able to adapt to a changing environment, okay? It says here, asexually reproducing organisms depend on mutations to generate variation, right? They don't have processes like this that are going to induce a large amount of genetic variation. The only variation that would come about by an asexually producing organism is really just simple, genetic mutations, okay? Meiosis, the two ways that genetic variation occurs in meiosis are in crossing over. Okay, so that's what I showed you. I'm going to show you more specifically those homologous pairs lining up and then exchanging genetic information. And then the other one is an independent assortment. So this has to do with whether the one cell gets the maternal copy of chromosome 1 or it gets the paternal copy of chromosome 2, and then chromosome 1, so what those combinations are. And I'll show you a picture and explain that as well. Okay, so like I said, the crossing over part, right, again, is exchange of genetic material between those homologous pair, right? Between non-sister chromatids. Because remember, the sister chromatids are exact copies of one another, okay? So again, this happens at synapsis. Synapsis is when those homologous pairs line up with one another. And they line up exactly so that they are just exchanging different forms of those genes, right? They're not going to, it's important that, you don't end up missing a gene or something like that, right? Okay. All right, again, the crossing over is the exchange of genetic material between those homologous pairs, not between the sister chromatids. That would induce no genetic variation because they are exact copies of one another's. The homologous pairs have the same genes, but remember, different variations, different alleles of those genes. Okay. And then it says the homologs separate and then are distributed to different cells, right? So again, this is actually an electron micrograph that's sort of showing you this lattice of proteins that essentially lines these up. Okay, don't worry. we're not going to get into details about this protein lattice here, but essentially that's what's going to make sure these homologous pairs line up exactly with one another. Okay, and now what do you notice happens? You have this crossing over that occurs, right? And if you look at these daughter chromosomes, they're mixed up, right? This isn't all of the paternal, it's got some maternal in there as well. Okay, so this lends lots of genetic variation, right? The other thing that we mentioned was independent assortment. Okay, so this has to do with where they go, what daughter cells get what chromosomes. Okay, so it says here, when homologous chromosome pairs align at the metaphase plate, right, they just separate in a random manner. Okay, so again, the maternal or the paternal homolog, I'm going to show you a picture in a second, may be oriented towards either pole, right? And then again, depending on which way they're facing, that's which way they're going to go, okay? And so again, you just get random mixing of these alleles, okay? So it says the possible chromosome orientations for a cell with 3 pairs of homologous chromosomes. Remember, we have, right, 23 pairs in our cells, is 2 to the 3rd or 8 combinations, okay? So you're going to see you have obviously a lot more different possible combinations in our cells, okay? So I'll show you what I mean here. So remember, during meiosis 1, those homologous pairs just line up at the metaphase plate here, okay? And so if they're lined up this way, this means this daughter cell that's going to be produced over here will get these, right? And this daughter cell will get these. However, if they're lined up differently, right, this daughter cell will get these and this daughter cell will get these. So if you think about basically in combination 1, and I'm not going to go through every single one, but you can look at them, each one will produce slightly different daughter cells in terms of whether they got the paternal or the maternal copy of the chromosomes. Okay, so here in this case, right, and sometimes it might help to actually kind of like draw this out. Usually I will do this on the board. I'll draw a cell, right? So if you have combination 1, tell me what those 2 daughter cells will have, right? So in fact, again, this daughter cell, so if we call this chromosome 1, chromosome 2, and chromosome 3, right? This daughter cell that will be produced, say, over here, will have all of the maternal copies of chromosome 1, 2, 3. This one will have all paternal copies of 1, 2, and 3, okay? In this case, combination 2, if we draw, we'll do it down here, we draw this daughter cell over here, this will have a maternal copy of chromosome 1 and 2, but a paternal copy of chromosome 3. On the other hand, this one will have a paternal copy of chromosome 1 and 2, but a maternal copy of chromosome 3. Okay, and so again, you can do the same thing with all of these, looking at the different ways these are lined up. And remember, in our cells, it's not just 3 pairs, it's 23 pairs, okay? So even more possible combinations. All right, so we talked about the two main ways that genetic variation is introduced in meiosis. It's during crossing over and independent assortment, okay? All right, so now a little bit more about genetic variation, because when we talk about sexual reproduction, it's a male and a female gamete that's going to come together. When we talked about meiosis, right, that genetic variation is just in the process of producing either the egg cell or the sperm cell. Now, when they come together, right? Depending on which egg cell and which sperm cell are joined, there's even more variation, okay? So it says in humans, there's 2 to the power of the 23 squared different zygotes possible. The squared part comes with the egg and the sperm coming together. The 2 to the 23, right, has to just do with this, the independent assortment, right? Okay, so what is this? I don't even know what that number is. 2, 3. What's this? This is million. This is billion, trillion, right? 70, whatever, trillion, over 70 trillion, okay? If crossing over only occurs once, then you're increasing this a whole bunch more, okay? And crossing over doesn't only occur once. It may actually occur several times on each chromosome, okay? So you can see how much variation is introduced just by these two processes that we talked about, okay? All right. So again, this is reiterating this, right? In asexual reproduction, you have genetically identical clones, right? Sexual reproduction, you have genetic recombinations, right, that occur. We just talked about kind of how much variation there is. It says here, in humans, right, the possible chromosomal combinations is 2 to the power of 23, okay, assuming no crossing over has occurred. But again, remember, then you're going to square that number, okay, to get this if we're talking about the joining of the zygote. So asexual reproduction is advantageous when the environment is stable, right? If there's nothing changing in the environment, then sure, you can quickly reproduce and have no genetic variation and they'll all be fine. However, if that environment changes, right, you would want to have genetic variability amongst those individuals of the population because some of them may be more suited for that new environment, right? If the environment changes and that whole population is all the same, they can all get wiped out by that environmental change, okay? So if you think about bacterial cells or something like that, right? If there's something changing the environment, it wipes out that entire population, it's gone, right? However, if there were some bacterial cells within that population that had slight genetic variations or genetic mutations that allowed them to be better suited for that new environment, they'll survive and then they'll produce more, okay? So genetic variation, no matter what type of organism you're thinking about, is super important. So I guess it's important to understand this concept. This could definitely be something that I might ask you about on an essay question, right? And really looking for you to answer it in your own words, right? Why is it so important to have genetic variability within a population, okay? All right, it says here, for an example, if the temperature rises due to climate change, right, an animal with less fur or reduced body fat would have an advantage. So again, you can think about it within an animal population. If every single individual in that population had a lot of fur and really high amount of body fat, they may all be wiped out, okay? All right, now we're gonna go through the phases of meiosis. And like I said, I may sort of kind of quickly go through these because you can look at the pictures, read what's happening at each phase on your own. I don't necessarily have to tell you all of this, and you will work on this, like I said, in your lab, okay? So I may sort of quickly go through this, so I'll just apologize, I guess, in advance, but there's a lot of important stuff to cover in this chapter. All right, so for meiosis 1, you're going to break meiosis down in those same phases that we broke mitosis down in, okay? So if we talk about meiosis 1 and we look at prophase 1, there's going to be a lot of things that are similar to prophase and mitosis, okay? So that's the other reason why I sort of usually go through this kind of quickly, because there are a lot of similarities, but also some differences. One thing that is different in prophase 1 of meiosis 1, right, is that this is where you're going to have that crossing over occur, right? Remember, the homologous pairs will line up. We call that synapsis, and then you will have crossing over, okay? This is just some terms here. It says synapsed homologs are referred to as a bivalent, right? Meaning those two homologous chromosomes together, or a tetrad, because there's technically 4 chromatids there. Just so you know, just a little terminology. In metaphase 1, the homologous pairs will line up at that metaphase plate, right? And then during anaphase 1, the homologous chromosomes, the pairs, will separate. Okay, they'll move to opposite poles. Remember, we talked about the independent assortment. That's happening there. the independent assortment is dependent on how the homologous pairs line up at that metaphase plate during metaphase 1, okay? Remember, during anaphase 1, the sister chromatids do not separate, okay? It's just the homologous pairs that separate, right? And again, at the end of, at the end of meiosis 1, you have haploid cells, okay? All right, so telophase 1, you basically have the formation of those two daughter cells, okay? Then there's something called interkinesis between meiosis 1 and meiosis 2. There will be no DNA synthesis or replication of the DNA, okay? It's just shorter and there's no DNA replication. So maybe those cells maybe grow in size slightly or what have you, but there is no DNA replication. And basically they're sort of the cells, think about the cells as kind of just getting ready for that second division, okay? Let's skip this for a second. I'll come back to that if I want. Let me just see something. It's kind of thrown in there in the wrong spot there. Yeah. You know what? Without the rest of the article and the pictures, it kind of loses its bite here. So within all the chapters, just so you know, there are little I don't know, they're kind of like short summaries of maybe an article or something like that. And a lot of times I'll print them out, we'll read them in class and we'll sort of go over it that way, or we'll do some sort of group activity with it. So anyway, this is just an overview of something that was presented in this sort of nature of science. Okay. So it's just giving you some explaining what this parthenogenesis is. Okay, so it's only one parent that contributes to the genetic information to the next generation. So if you're interested, like I said, it's in your textbook on page 171. You can take a look at that. All right, sorry, that was sort of a little side note interruption. So we went through meiosis 1, now we're in meiosis 2. And essentially, like I said, meiosis 2 is really like mitosis, right? So prophase 2, the chromosomes condense. They will, in metaphase 2, they'll align at the metaphase plate. Now they will set, those sister chromatids will be separated during anaphase 2, right? And then telophase 2 and cytokinesis will produce those 4 haploid cells, okay? And remember, these are all genetically unique. These are not genetically identical. So mitosis produces 2 genetically identical daughter cells. In meiosis, if we're going through meiosis 1 and meiosis 2, you end up with 4 genetically unique daughter cells, okay? Gametes. All right. So like I said, you can look at these stages again. I sort of just went over them. But if you look at, again, meiosis 1, right? And then meiosis 2 here. So remember, these 2 daughter cells are produced at the end of meiosis 1. And then each one of those will then divide essentially, okay? And remember, it's sort of, this is showing you here in prophase 1, you can see this crossing over that's occurring, right? This is that genetic variation, one thing that will lend to more genetic variation. And metaphase 1s, right? Remember, metaphase 1, it depends on how these homologous pairs line up, right? This is the independent assortment that we talked about. Also notice that because of crossing over here, you see these homologous pairs, right? There's some exchange of genetic variation between the maternal and the paternal copy, all right? Okay, so again, I already just went through all of this. This picture, shows you what it looks like, points out some of the main things that are happening at each phase. Again, remember, meiosis 2 looks really similar to mitosis, all right? Okay, if you want to just look at it, and this is a little bit bigger, so it allows you to see it a little bit better. All right, so again, this is showing you telophase, okay? And then obviously cytokinesis, remember that cleavage furrow that forms, and we have the pinching of those cells to produce those 4 daughter cells. Okay, so that's what this is actually just showing you a picture here, right? Of the formation of those 4 gametes. Okay, so I've sort of been doing this all along, but we want to be able to make sure we can compare and contrast meiosis and mitosis. So like I said, I've probably said all of these things as I was going through. Okay, so this again just sort of summarizes that. Meiosis, 2 nuclear divisions. Mitosis, only 1. Here in meiosis, you're going to have the chromosomes synapsing and the crossing over occurring. You don't have that in mitosis. Okay. Here in anaphase 1, another thing to point out is that in meiosis, the centromeres stay around. Why? Because you're going to have another division that occurs. Here in anaphase, the centromeres will dissolve. Okay. All right, so anyway, I think I kind of went through all of this already. I don't know that I need to read it again. Make sure we know the differences between meiosis and mitosis. Okay, so this is pointing out really the differences. Remember, there are many similarities as well. So a lot of times I do ask you on an essay question, list 3 similarities between meiosis and mitosis, and list 3, differences between the two. All right. Okay, so again, those were differences. If we look at similarities, they're listed here. Okay? I mean, I think the similarities are pretty obvious. Okay? Again, there's another table there comparing meiosis. So this is comparing meiosis 1 to mitosis. Okay? And then this compares meiosis 2 to mitosis. Again, I pretty much pointed out most of these things as I went through describing the stages of meiosis. Okay? Again, in picture form, right? Comparison of meiosis versus mitosis. Right? So again, make sure you can pull out similarities and differences between the two processes. If you really understand the two processes, it's easy to do this, even without having these slides, right? If you understood the two processes, you could pull out these similarities and differences yourself. Okay? All right, so we are going to, I don't know why this is not letting me move here. Okay, moving on, right? So this next part is pretty short. It's just kind of going over the cycle of life and kind of thinking about where meiosis fits in to the life cycle, okay? And where mitosis fits into the life cycle of a organism, all right? So it says here, the life cycle is all the reproductive events that occur from one generation to the next generation. Okay, that's that life cycle. If in plants, okay, there are actually haploid individuals that exist, and you can it's kind of like an alternate version of this sort of the sort of bigger plant. A lot of times these haploid individuals are smaller versions of the plant. Okay. Anyway, the sort of haploid and the diploid individuals you alternate between them, and you call this alternation of generations. So that's something that's specific to plants. We're not going to talk about it right now. But it's a term worth knowing. Okay. And so again, within this, right, we call the haploid individual, okay, a gametophyte. Okay, so it says it may be larger or smaller than the diploid. A lot of times it's smaller, but it depends. Depends on the plant you're talking about. The diploid is called a sporophyte. Same thing. It can be smaller or larger depending. And so again, you only see this alternation of generations in plants, and actually I don't know that that's true. I think there are also some protists that might display the alternation of generations. But for the most part, when you think about alternation of generations, you think of plants. So here it's giving you some examples here. Like mosses are haploid for most of their life cycle. So they don't have a large or a substantial diploid phase. Okay. Fungi, algae, only the zygote is diploid. Everything else, they're haploid. Okay. Ferns, higher plants are diploid for most of their life cycle. So there's variation within the plants. Okay. All right. Anyway. So like I said, we're not really going to focus too much on this. I guess for this, the only thing you really need to know is really what is meant by alternation of generations, and that we see it mostly in plants. Okay. In animals, right, we'll spend a little more time on this, but not a lot at all. It says individuals are diploid, right? So if we talk about humans, we talk about a dog, they're diploid, okay? The only thing that's haploid are the gametes, right, inside you. Those egg cells are those sperm cells, okay? So that's what this says here. The products of meiosis are always gametes, okay? So it says meiosis occurs only during gametogenesis. I said this when I started talking about meiosis, okay? The only time meiosis happens is when you're forming gametes, okay, in your body. So production of sperm we'll call spermatogenesis, okay? All 4 of those cells that are produced become sperm, okay? In oogenesis, production of eggs, it's different, okay? One of the 4 nuclei actually will get the majority of the cytoplasm and become the egg. The other ones just get discarded, okay? So the meiosis process sort of still occurs the same way in terms of that nuclear division and the formation of those 4 nuclei, but only one of them will become a viable egg, okay? So you can see how those are different. If we talk about the life cycle, right, I think I already kind of said most of this. Sperm and egg are produced by meiosis, right? Sperm production, spermatogenesis, oogenesis, egg. Know those terms. Again, I don't really hold you responsible for knowing exactly what's going on, but really spermatogenesis is meiosis, right? It looks just like meiosis. In oogenesis, there's definitely variations, but that's something that you would go over in more detail, maybe even in anatomy and physiology too, also in like a developmental biology course. So I don't focus too much on the subtleties or the specifics of those 2 processes, okay? All right. So it says sperm and the egg fuse at fertilization. We talked about this already. Results in a zygote, okay? That zygote then goes through lots and lots of mitosis, right? And then goes through that developmental process to form an embryo and then a fetus, okay? So that's what this is saying. The growth is mitosis, right? All right. Anyway, I think I already said all that. Okay, it says here, as a result of mitosis, each somatic cell, so that's the rest of your cells in your body, right, has the same number of chromosomes as the zygote and has the same genetic makeup as the zygote. So if you think about the cells in your body, every single cell in your body has the same DNA, has the same genetic makeup, okay? Now I always have to say there's a caveat there. So there are immune cells where you actually have some DNA rearrangements that go on, but that's when we're talking about production of cells that produce antibodies and stuff like that. But for the most part, we're going to say every single cell of your body has the same genetic makeup. What makes a lung cell different from a brain cell is the different types of proteins that are produced. Okay, it's not that there's different genes there. It's because those cells produce different proteins or express different proteins. Okay, so that we talk about a little bit more in chapter 12. All right, so that's a little bit of a preview into that chapter as well. Okay, so like I said, you guys don't need to know the details of spermatogenesis and oogenesis. I just want you to know that spermatogenesis is the production of sperm. It takes place in the testes, obviously only in males. Oogenesis is going to take place in the ovaries and is the production of egg cells. Okay, and you can just note some of the different the differences here. But again, you don't really need to focus on this and know all the details. Okay, I'm just going to pull up the picture here. Right, so the one thing to note, and I'll show you in a picture too. The secondary oocyte begins meiosis 2, but actually stops at metaphase 2. Leaves the ovary and enters the uterine tube. So if it doesn't get fertilized, it will degenerate, okay? If it does get fertilized, it will trigger that secondary oocyte to complete meiosis 2. So it halts in metaphase 2. It only will complete metaphase 2 if it encounters a sperm. Okay? So if you look at the life cycle here, right? Again, we're showing you that these are 2N individuals, any individual, right? 2N. And if you're talking about going from, a baby or even going from a fetus to an embryo, right? Or an embryo to a fetus, sorry, and then to an actual fully developed baby, this is all, and the growth that occurs from infant to childhood to adult, it's all via mitosis, okay? The only thing that meiosis is involved in is the production of egg cells and the production of sperm cells, right? Once that zygote forms, it's all mitosis. All right, so this is basically showing you what the role of meiosis is and mitosis is in terms of the whole life cycle of a human. All right, sorry. So I think we should already know that. All right, and like I said, you're not responsible for the details of spermatogenesis and oogenesis. Let's just know what they are. All right. Okay, finally, the last part of this chapter, I believe this is the last part, is to talk about changes in chromosome number and structure. Okay, so what happens if things go wrong during meiosis? Okay, and for the most part, it usually occurs normally, right? However, there are instances where it does not occur normally and there are problems. Okay, so some terms here. Euploidy is a term that means is a term for the correct number of chromosomes in a species. Okay, if we talk about aneuploidy, it's a change in chromosome number, whatever change that might be. Say some change. All right, and we're going to talk about some of these things. Another term here, karyotype. I mentioned this in the beginning of the chapter. I showed you a karyotype. Basically, it displays all those chromosomes. They're arranged by size and shape. You can see the banding pattern, and you can look for differences in the number of chromosomes from that karyotype. Okay, so if there is some suspicion that there might be some sort of chromosomal abnormality, you basically, a geneticist can look at that karyotype to see if there's an abnormality there. Okay, this really only just shows you changes in chromosome number, okay, and structure as well. It's not going to show you a single mutation of a gene or anything like that, but it will show you changes in the chromosome. Okay, so it says aneuploidy, right, results from non-disjunction, which is the failure of chromosomes to separate, right, during meiosis 2 or meiosis 1. So basically, again, you'll see that chromosomes, instead of, one going to one daughter cell and another going to the other daughter cell, they're going to both go together, and then one daughter cell might not even get that chromosome. Okay, so it leads to too many in one daughter cell and not enough in the other. All right, so I'll show you a picture that kind of shows you this non-disjunction and how it occurs and what might happen. So like I said, it may result in the gain or the loss of chromosomes. Okay, there's some more terms here. Monosomy is a term that describes only one of a particular type of chromosome. So this means, say, chromosome 2, you only have one copy instead of two. If we talk about trisomy, you have 3 of a particular type of chromosome. So instead of having 2 copies, you have 3, an extra copy. Okay, both are dangerous. All right, so this is going to show you this non-disjunction. Okay, so we said that aneuploidy will occur due to non-disjunction during meiosis, okay? So it's showing you, this is kind of showing you 2 different examples. So this is non-disjunction that's occurring here, right, in meiosis 1. So here's your pair, and again, it's oversimplifying, just showing you one pair here. In this case, instead of one going here and one going here, both went here, this got none, okay? Then when it goes through meiosis 2, right, these are gonna, right, separate and move here. Again, you got nothing here. This is in terms of just showing you, again, just one example of this happening in one pair, right? And then what do you end up with here? You're missing, right? You're missing a lot of chromosomes here, okay? Once that fertilization occurs. Okay, so this is showing you basically kind of, well, I mean, whatever. Let's just, yeah. So this is sort of, right, because it's not necessarily kind of showing you the production of the egg cells, right? But remember, you don't really actually produce 4 viable egg cells anyway. But regardless, this is just showing you hypothetical situation where this might have happened in that one, what's going to become that viable egg cell. And again, depending on what one was fertilized, right, you're going to end up with a different type of issue, okay? So in here, you have extra, right? You got extra. Here you're missing, right? So you have two copies of that maternal chromosome here when you're only supposed to have one, right? Here you don't have any copies of that maternal one, so you're missing, okay? Non-disjunction is occurring at this point here where now instead of one of these going here, one of these going here, 2 went over here, right? And you got nothing here. And again, what you're going to end up with is, in this case, when it happens in meiosis 2, you can actually end up with a couple normal ones, right? A normal zygotes, okay? And then these are going to be abnormal, okay? If it's happening in meiosis 1, it's going to lead to all these zygotes being defective, okay? So anyway, I just wanted to illustrate kind of where, where this could happen, and this could happen in, there's obviously many other sort of possibilities of issues that occur. This is just giving you some examples here. We said that you get an abnormal number of chromosomes because of non-disjunction. And so basically this is chromosomes not going to where they should go, right? And sort of getting stuck together here, and then again, you end up with a cell where you don't have anything, right? And a cell that has too much, okay? So hopefully that makes sense. So this is, again, just giving you two examples here of non-disjunction occurring at different places during different stages of meiosis, okay? And what then is the end result? All right. So we said trisomy, right, occurs when an individual has 3 of a particular type of chromosome, okay? It says, so in humans, there is actually only 3 autosomal trisomies that are viable beyond birth. So there are many other trisomies that might occur, but these fetuses are not, or, these offspring are not viable beyond birth, okay? So that means you would basically, give birth to a non-viable fetus, okay? One that we probably know about and heard of, the most common one, is trisomy 21, right? So you have an extra chromosome 21. It is called Down syndrome, okay? And the chance of a woman having a child with Down syndrome is believed to increase with age, because the longer those oocytes are stored and are around, because remember, you produce those, early on in life, right? And then they're there. You have a greater chance of non-disjunction occurring, okay? Some characteristics of Down syndrome: short stature, eyelid fold, right? These are the sort of characteristic, or the physical characteristics. When you look at a patient with Down syndrome, you usually can pretty quickly recognize that they have Down syndrome, okay? Flat face, stubby fingers, wide gap between the second and first toes. If you look at a karyotype from an individual with Down syndrome, what do you notice? If you look at chromosome 21 here, there are 3 copies instead of just 2, okay? This is particularly showing you that this one gene that exists on this chromosome, don't worry about that, okay? This GERT gene, it says. So, yeah, so I guess it's believed, or there's been studies that have shown that this one particular gene, and it's not only this gene, but that this is one of the genes that's a major player in terms of the many of the characteristics that you see in Down syndrome, because basically having an extra copy of this gene, right, because you have an extra chromosome, produces really high levels of purine in the blood, and apparently that has to do with some of the characteristics of Down syndrome. But remember, again, this is not a genetic disease that's caused by a single gene mutation here. This is caused by a whole extra chromosome. So there are many other genes on this chromosome as well that you're going to have extra copies of, and therefore be producing, extra of certain proteins that then can lead to problems, right? So more is definitely not better in biology, okay? Remember, we always have to maintain homeostatic levels of proteins and molecules within the cell, right? So if there's something that's going to cause more of a molecule to be produced, a lot of times that will be dangerous to the cell and to the functioning of the body as a whole. Okay, now, so again, there are not many autosomal trisomies, so meaning trisomies of the autosomes, which are all the chromosomes that are not the sex chromosomes, okay? So many of these produce non-viable offspring, okay? And so that's why we're only mentioning trisomy 21 here. There are here it says 3 autosomal trisomies that are viable beyond birth. We're only going to mention trisomy 21, but there are not many, okay? Because again, having an extra chromosome, especially, of an autosome, an extra autosome, is going to be very dangerous, okay? However, if you have changes in the sex chromosomes, okay, it's more tolerable, okay, because there's not as many genes there, I guess, if you want to think of it that way, I guess. Okay, so results from inheriting too many or too few X or Y chromosomes. Okay, so again, I just told you that extra copies of the sex chromosomes is definitely more tolerable than those autosomes. Okay, and again, this is going to happen because of non-disjunction. Some examples of disorders caused by a change in number of the sex chromosomes. One is Turner syndrome, okay, where you have an XO. So it's a female with a single X chromosome, right? So instead of normally a female will have two X chromosomes, right? In this case, you only have one X chromosome, okay? So here you're going to have some physical sort of, it's not an, I don't know that it's abnormalities or sort of characteristics, sort of physical consequences because of this, right? Short stature, the chest is more broad. But most of these, or a lot of these patients would be normally intelligent and they would just need to take hormones, right? So estrogen replacement to sort of make up for the lack of those female hormones, okay? And so for the most part, I think can function pretty well, okay? If we talk about another syndrome called Klinefelter syndrome, here you're a male, right? Because you contain the Y chromosome. So wherever if you have the Y chromosome, you're considered a male, okay? So in this case, the male has two X chromosomes and a Y. So normally a male would have one X chromosome and one Y chromosome. So in this case, male has underdeveloped testes, right? And prostate, some breast overdevelopment, again, because of the presence of these two X chromosomes, long arms, long legs, large hands, near normal intelligence. Again, it's not going to really affect the nervous system or the intelligence level. However, if you start to get to cases where you have three X's or four X's and a Y, then it's going to start affecting intelligence and brain function, okay? I already mentioned this, that again, wherever there's a presence of a chromosome Y, that renders the individual as a male, okay? And it's because of the presence of a certain gene on that Y chromosome, okay, which is this SRY gene. So in fact, if you just delete that SRY gene, right, from the Y chromosome, basically it ends up, because it lacks this testes determining factor, which plays a major role in the development of the male genitalia, this results in basically a female because the testes do not form. Okay, so it's really believed that this SRY gene, which is present on the Y chromosome, is really the determinist of, or really determines maleness, whether you're a male or not. Okay, and most of it has to do with its role in the development of the testes. Okay, so again, some more karyotypes, right? So we should be familiar with looking at these. And so if I showed you a karyotype on the exam and ask you, you know, does this individual have Turner syndrome, Klinefelter syndrome, Down syndrome? Is it normal? We should be able to determine that by looking at the karyotype. So in this case, again, if we look here, we see there's a missing X chromosome, right? So this is, we would term this individual as XO, right? Has one X chromosome, but is missing the other. So a female normally has two X chromosomes, right? A male normally has an X and a Y. In this case, you have two X's and one Y. Again, the presence of the Y chromosome, if you see the Y chromosome, you would then determine that this individual is a male. Okay, so again, if I ask you from a karyotype, is this individual a female or a male, we should also be able to determine that as well. So remember, you have 22 pairs of autosomes and then one pair of sex chromosomes, okay? So again, male, an X and a Y, right? In this case, you have an extra chromosome X, which is characteristic of Klinefelter syndrome. In females, normally you have two X chromosomes. In Turner syndrome, you're a female, but you're missing one of those X chromosomes. Okay, now that was basically talking about changes in chromosome number. Okay. This next part talks about more about changes in chromosome structure. Okay, and basically changes in chromosome structure. Okay. It says here, environmental agents like radiation, organic chemicals, or certain viruses can actually cause chromosome breakage. Okay. If those broken ends of those chromosomes don't rejoin where they're supposed to, right, or right where they broke, then it's going to cause mutations. Okay. So here are some changes in chromosome structure that can occur. One, deletion. Okay. One or both ends of a chromosome breaks off, and that's it. It's lost. Okay. If you have two simultaneous breaks, it can lead to loss of an internal segment. Okay. So, something breaks off, it could just be deleted altogether. There's also something called duplication. Okay. So there's a presence of a chromosomal segment more than once in the same chromosome. I'm going to show you pictures here. So in this case, you're not missing anything, but you have a part of the chromosome that gets duplicated. Not the entire chromosome, just a segment of it. Translocation, a segment from one chromosome will move to a non-homologous chromosome. Okay. It says it follows breakage of two non-homologous chromosomes and improper reassembly. So this is like two parts break away, right, from two separate chromosomes, and then they reinsert in the wrong place. Okay. Last one, inversion. So you have two breaks in a chromosome. That segment reverses or inverts before it gets reinserted. Okay. And so then you have genes that are occurring in an improper order in that inverted segment. So let me show you pictures here. And then I'll go back to those syndromes in a second. So deletion, right? You can see here, you're missing this end here. A lot of times it's the ends of the chromosomes that will break off. Duplication, right? You have this segment that's actually duplicated here. You can see that. Inversion, right? You can imagine that maybe this part of the chromosome broke away and then it got inverted before it was reinserted. In translocation, now you have parts of two different chromosomes breaking off, and then they went back into the wrong places, or they basically reattached onto the wrong chromosome. Okay? So these types of abnormalities in terms of changes in chromosome structure can lead to syndromes, right, and diseases. Okay? So these can be detected by karyotype as well, also by looking at inheritance patterns in families. Examples of deletion syndromes, okay, Williams syndrome is one, where you have loss of the end of chromosome 7, okay, and particularly one of the major genes implicated in this disease or syndrome is the gene for elastin, okay. But there's also going to be a lack of other genes that are present in that area as well. So these children have a characteristic sort of physical appearance where they look like pixies, but they also have problems with mental function as well. Okay. So in terms of, it says their academic skills are poor, but they do exhibit excellent verbal and musical abilities, right? So that's something that sort of makes them unique. They also have some other health effects because of that lack of elastin that I mentioned there, in terms of cardiovascular defects, and also it causes their skin to age prematurely. Okay. All right. So that's one example of a deletion syndrome, Williams syndrome. All right. Translocation caused syndromes. So Alagille syndrome is Largile. Actually, I think it's Largile. I gotta look up that. I don't know why I'm like mispronouncing it though. But anyway, I'll probably do a little check on my pronunciation of that, and I'll correct myself on Tuesday during our meeting. But anyway, this syndrome is, I have like a little brain fart there. So this syndrome is a translocation between chromosome 2 and 20, and it leads to this particular congenital heart defect that's called tetralogy of Fallot. And basically, it's this sort of, there's 4 characteristic heart issues that occur. Okay, so that's where this name comes from. And that's part of this syndrome here. Okay, and again, there's a translocation between chromosome 2 and 20 in this case. A type of leukemia, chronic myelogenous leukemia, which is a blood cancer, is caused by a translocation between chromosomes 22 and 9. So another example of a syndrome that's caused by translocation. Okay, so if you look at this, this is the Williams syndrome, right? If you look at these children, they all have a sort of similar characteristic appearance in terms of kind of the shape of their nose, and also they sort of have wide mouths, large ears, and a small chin. And so that those physical characteristics are the same. So the point of this picture is to show you that these are not siblings, but they really do look alike because of having this Williams syndrome. Okay. Here, this is actually showing you an example of see that I knew that I think this is a typo. Yeah. Anyway, let me just correct it now so that you see that. It's allogeal, I believe. That's why I was like, what is going on here? Where is that? Okay, so I'll just double check that I'm correct with this, but it is I believe that's a typo there. Okay, so I'll correct that. Well, I just kind of did correct it, but I'll just double check. I'll let you know if for some reason I corrected it the wrong way. Okay, so it's allogeal syndrome. Okay, that's why again that was throwing me off because it's a typo there in terms of the spelling. So you actually have sort of these sort of club fingers that are characteristics of this as well. So like, if you went to a geneticist or a genetic counselor, right, and they saw, or a clinician, right, and they saw this sort of characteristic clubbing of the fingers, it would indicate to them that potentially you might have this syndrome, right? So that's why it's important to point out these physical characteristics, right? Because these are things that will indicate to the clinician that it could be this genetic disease or a genetic syndrome. Okay, so that's why we mentioned those physical characteristics. All right. So again, this is caused by the exchange or translocation between chromosome 2 and 20. Okay. And again, you have these distinct body features, but also have heart issues as well. Okay. So again, these were just meant to give you some examples. I may ask you to give me an example of a deletion syndrome or a translocation syndrome, but I think for us, we want to just make sure we understand the different types of chromosomal changes in chromosomal structure that can occur. Okay, and so we only gave a couple examples of syndromes that can be caused by changes in chromosome structure. There are many others as well. All right, and again, I guess another sort of take-home message is to understand that all of these sort of genetic abnormalities or syndromes or diseases that are caused by either a change in chromosome number or a change in chromosome structure have these characteristic physical attributes that help clinicians identify a patient as maybe having one of these diseases. Because remember, some of the symptoms might be shared with other types of diseases as well. All right, guys, so that concludes chapter 10. The next chapter is chapter 11, which is really the genetics chapter, where we will talk about alleles and homozygous and heterozygous and those terms a lot more. We'll also talk about Mendel and his contribution to genetics. We'll do some crosses and things like that. So just a little bit of a preview to the next chapter. All right, thanks everyone.